Well, the one thing I wanted to finish up on was the work, and we've talked about this, mentioned this a couple of times already, and Dave, you might like to add some comments to this as well. So we have these microsatellite markers with rust that we can now use to do genotyping, and we've found that there's variation within pathotypes. Dr. Szabo, Les Szabo, who some of you may know in the US, has actually developed SNP markers for stem rust. And these are proving to be particularly powerful in discriminating not just whether or not something belongs to the UG99 lineage, which is illustrated here, one of Dave's diagrams here showing the different variants of the UG99 lineage and their distribution. This is out of date, but when would this have been, Dave?
2011.
2011, so it's a little bit out of date. But anyway, there are variants within UG99, and we believe they're single-step mutants. Les has now developed SNP markers that are allowing him to discriminate within the UG99 lineage, and not with 100% confidence, but with quite a high degree of confidence, maybe 90% plus, actually say, on the basis of a SNP genotype, it's this pathotype. And that's pretty amazing. So this is what he does, and Dave has run you through this. All it takes is a piece of leaf like that with a single pustule, right? They do a total DNA extraction on that. So the DNA they're using to do the genotyping has plant DNA in it and rust DNA in it, but the markers are specific for the rust. So the fact that the plant DNA is there doesn't matter. The markers are only specific, the primers only go and stick to the rust DNA. So from a single pustule, Les is able to run his SNP markers, and this is just an example of 1, 2, 3, 4, 5, 6, 7, 8 of those SNP markers, and you'll see the different alleles. They're either the T or the T's or the A's, or the C's or the T's. And on that basis, he can actually identify different pathotypes. So in this case here, the first one is TTKSK, and in fact, and this may have changed since this data, but he was only able to find one genotype for that pathotype when he looked at multiple isolates of it. So this particular genotype here for those 9 markers, or however many it is diagnostic, you could say, for race TTKSK. So when he sees this genotype, he would say from that single pustule, this isolate is probably TTKSK without doing any seedling tests in the greenhouse. The one down here, where I have the 2 arrows, is actually TKST, and he's found 2 genotypes within that pathotype. So here's an example where we have one pathotype but 2 different genotypes. All right? So that then makes it a little more difficult to actually predict the pathotype. But anyway, just an example of, I'm not too sure how many, but quite a few, and Les has done a lot more than this now, but these were quite a few single pustule samples, Dave, that I would imagine you sent to Les back in 2006 possibly, no, 2011. And based on that SNP genotype, Les has predicted the pathotypes. And I think they then did the phenotyping in the greenhouse later. Dr. Eugene did that and pretty much found there was 100% correlation. Is that right? Yep. Yeah. Do you want to add anything to that? I mean, these are slides from 3 or 4 years ago, and Les has actually developed this to an even further refinement now.
Yeah, I mean, what Les is finding now, I mean, an increasing number of different genotypes within the same race phenotype. So I think when I last saw Les a few weeks ago, I think he had 18 different genotypes within race TTKSK. So there's a lot of variation there is what's coming out. And he's also, I mean, the assay is developing, so he can now look at other important races. So the Digaloo race, TKTTF, his assay is now diagnostic for that race. I mean, that was an interesting one, that in Kenya on Robin, which we knew was carrying this TMP gene, all the DNA assays were telling us it was TTKSK, the original UG99. And it wasn't making sense to us at all. But his assay wasn't discriminating between these, what we now know are new variants of UG99. So we've now got the TMP variants of UG99. But that his assay, I guess, was it was an interesting result, that it clearly, it wasn't the race we had in Ethiopia that was virulent on TMP. So that was our first sign it was something different in Kenya from the DNA result. But he couldn't discriminate with the DNA assay. So that's an indication that it's not perfect. Yeah. But even so, the result was interesting. And then subsequent race analysis, we now know we've got actually 2 UG99 variants that are virulent on TMP.
Yeah.
But that was the first clue that it was. We assumed originally it was, oh yeah, it's what we've got in Ethiopia. It's just moved to Kenya. But that was incorrect.
Okay. Better pull it up there, I think. Yep.
Do you have any other-
That's it.
That's it?
Yeah.